This is passage 3 of the AAMC section bank, and this is a volume 2, and then this is the biochem section, and then this is the passage right here. Human immunodeficiency virus, or HIV, is a diploid retrovirus that, if left untreated, will lead to acquired immunodeficiency syndrome, or AIDS. HIV is characterized by the targeting and destruction of CD4 plus T lymphocytes, which results in immunodeficiency. HIV is selective for CD4 plus cells due to the specific ionic binding of its external glycoprotein, GB120, to CD4 receptors, thus initiating a viral injury. CD4 cells express another membrane co-receptor, or known as CKR5, that is needed for HIV entry. Research shows that people who are homozygous for a 32 nucleotide deletion results in 30 fewer amino acids in the sequence of CKR5 and are resistant to HIV. So something like important here, because anything shown research related, I like to kind of highlight. Once inside a cell, HIV replicates its genome and integrates within the host chromosome as part of the antiretroviral therapy, or ART. Nucleotide reverse transcriptase inhibitors, NRTI, and protease inhibitors are used to treat HIV infection. NRTIs are nucleotides that lack a hydroxyl group at their 3' deoxyribose and therefore cannot form a 3' to 5' bond. And the growing nucleic acid chain. Compound 1 is an HIV protease inhibitor that binds to the active site and inhibits the function. So gives a dissociation constant right here. The main obstacle to finding a cure for HIV infection is that the viral genome can remain dormant in infected cells. Because these cells do not express the HIV antigens, they are not recognized by the immune system and therefore are not targeted for elimination. Researchers have developed latency reversing agents, LRAs. I like anything that's coded for acronym, such as histone deacetylase inhibitors. These inhibitors prevent deacetylation of lysine residues and the chromatin-associated histones. So they're saying keywords here, acetylation and all that. Okay, I'm thinking right with DNA, like the type of like chromatin or urochromatin, thus stimulating virus production, resulting in viral elimination by the immune system. And that's passage 3. Question number 1. Based on the passage, CKR5 most likely undergoes which type of mutation? And so they talk about this in the second paragraph, where they say that those who are homozygous for a 32 nucleotide deletion results in 30 fewer amino acids in the sequence of CKR5. So then the best way to kind of go through this is kind of go the answer choices one by one. I believe this information will be enough to answer the question. So let's look at starting answer choice A, a missense mutation within the gene, intronic gene. So now, two things. One, they say missense. So recall that a missense mutation is when you change one part of amino acid. They're talking about like a 32 nucleotide deletion. And so like the 32 nucleotides are like removed. And so if there was like a missense mutation, I expect there to be like a change in like one type of nucleotide. And so for example, it can go from like a iodine to thymine. That's only being one chain inside the the mutation, versus as there's 32 nucleotides deleted. So right away, because of that, this is not a good answer so far. And then the second part of this answer is saying that gene intronic region. And then recall that introns, there are like the non-coding regions for DNA. They serve more as like a structural property rather than for coding proteins. So intronics don't really do anything, and so this has no effect at all on the final product of the protein. So A is not a good answer so far. Now let's look at B now. They're saying a missense mutation within the gene promoter region. Now, same thing right here as last time. Missense, they only changed the one nucleotide, and as we recall, there's 32 nucleotides deleted. So still, regardless again, a missense mutation is still not a good answer. And then the second part of this question answer is saying that within a gene promoter region. Now, if you were gonna change a mutation in the gene promoter region, it's the region where like we first start transcribing. So really, it's not gonna affect how we're gonna have the 32 nucleotide deletion, because if we put like a change in the amino acid, it's still gonna go on, right? It's not gonna like change out the outcome of like the 32 nucleotide deletion. So because of that, B is still not a good answer. Now, if you look at C now, it's a little bit different. They're saying now a nonsense mutation within the gene promoter region. Now, a nonsense is, if you recall that, it's a premature stop codon in which like it stops the transcribing of like for a gene. And so that's where kind of it's like a stopping point for it. So now, if it's in a promoter region, that means there's nothing like being produced at all. And then now they're saying right here in the sentence, they're saying that they're implying that resulting in 30 fewer amino acids. So they mean that the protein was already produced. And so, but the only difference is that the protein from the original is that there's 30 fewer amino acids. So they're implying that the protein was already made. This means that, like, in this answer choice, they're basically saying no protein was produced at all, because if you put a stop codon inside promoter region, the place where transcribing starts, it's not going to make a protein at all. So this doesn't make any sense. So C cannot be a good answer either. Now D, much better, promising answer. For one thing, a frameshift mutation could explain why there's 30 fewer amino acids. And so if I were to pull a diagram up right here, so let's say like this is our normal, and then now if you were shifted down this way, right, this could potentially go in a region where you know, you are in a you could go into an intron area. And so if you're like being frame shift down to an intron area, you can have less amino acids produced, because remember, intron areas do not produce amino acids, or they don't transcribe anything. And so this could possibly result why there could be fewer amino acids in the sequence. And so this will make the first part makes makes sense so far. And now the last part is saying the like exonic. So an exon, it's like, recall that that's where the protein, or like where the gene is like being transcribed. And so it's the one that like creates the protein or being translated into. And so this will make more sense than versus an intron, because introns don't do anything to create proteins, while an exon does, because during like splicing they remove all the introns and they're left with the final product, which is the exon, to create the protein product. And so D makes the most sense, and it is our correct answer. Question number 2. What is the function of the LRA described in the passage? So now they talk about LRAs right here. They say that these inhibitors prevent deacetylation of lysine residues in the chromatin-associated histones, thus stimulating virus production and resulting in viral elimination by the immune system. Now, they're talking about packaging of DNA, basically. So this is something you should recall for content right away. And so just recall that, like, for your sake of content review. Remember that histones, these are positive charged molecules. They're the result from lysine or arginine positive amino acid. And so this is what gives it its overall like positive charge. And now DNA, it gets its negative charge from like negative phosphate groups inside the DNA molecule. If you don't believe me, you can look it up. I recommend you know that. It's just a good something to know. And so because of these positive and negative charges, they like to interact with each other. And so when they interact with each other, the histone molecules become more coiled up with the DNA strand. And so when they're more coiled up, what do you think what happens? Transcribing decreases, because there's not enough space for them to like get transcribed out in the open in the cytoplasm, or like in the nucleus. So if like so then yeah, these be condensed close together. Now, whereas if we were to find a way to like somehow like disrupt these positive charges, then these like DNA strands can become loose, and then increase transcription. So that's what we need to know and go into this question. So let's go back to our question. And I like to go through these answers one by one, and I think a good shortcut is kind of like, if you really know your content well enough, you could notice that right away. It's like it says ear chromatin, heterochromatin, and they're even given like the little definition at the end of them. So if you recall that heterochromatin is something that it's closed or condensed, and it doesn't actively allow transcription, whereas erochromatin, it's like actively allows transcription because it's more open, and like the DNA strands are out more open. So we can actually eliminate like anything that doesn't match. So if, like, for example, like A, your chromatin says allows transcription factors. Okay, that makes sense. I'll keep that. And then I'll look at B. They're saying your chromatin prevents, but then we just said that your chromatin increases transcription. Why would it prevent it? So we can automatically eliminate B regardless if we didn't even read the passage or not. We know that this is a complete false statement. And then now if we look at C again, this says increases heterochromatin formation, thus allowing access to transcription factors. So like heterochromatin, remember, closing condense, decreases transcription. So this definition doesn't match at all. And it's wrong regardless if you even read the passage. So right away, we're already down 50-50. I haven't even read the and if we're taking the test, and I haven't even read this part yet, I'll right away like have two like answers eliminated. So now we're stuck between A and D. So let's look at A first. It's saying that it increases erochromatin formation, thus allowing access to transcription factors. So they're saying right here, the inhibitors prevent deacetylation of lysine residues. Now, this is a lot to like chunk in, like, it's a lot of wording right here. So, like, it might be a little bit confusing. So I'm just gonna kind of break this like piece by piece a bit. So what I like to focus on the word first is acetylation. Recall that acetylation is like when you're kind of like, I know there's like these special molecules they add to the thing right here, but I'm just calling pins for now for the sake of explanation to keep it simple. So bear with me. I'll just call these pins for the rest of this question. So these pins supposedly have a charge, a special charge that disrupts these like histone like positive charges. So if these are disrupted, that means you get less positive like histone octamer right here as you keep acetyling this area. And so if it's disrupted, that means it cannot bind well to DNA. And so what happens? It becomes loose. And so it uncoils and then DNA gets exposed. And so that's what acetylation is. Now look, deacetylation. Now this part right here, D means the removal of it. So basically, to put in simple terms, is basically the removal of these pins. And so when the pins are removed, you bring back the positive charge. Now, and it coils back up again. And now they're saying these inhibitors prevent the removal of these pins. So basically, they're saying that these pins are being stayed in place. They're staying there. And so that's what we have to know. So if we know they stay there, that means they're disrupting charge and then the DNA becomes loose. So that's how you kind of want to break it down if you're like so confused, like just by reading this sentence right here. And so we know that because of the pins are staying there, that means that DNA must be unwinded and the transcription must increase. And so A matches that because it's increasing transcription, and D is the opposite of that. So A makes the most sense, and then that is the correct answer. Question number 3. Based on the passage, NRTI prevents the formation of which bond? So they first talk about NRTIs in this paragraph right here. So let's read. They said they are nucleotides that like a hydroxyl group at their 3' deoxyribose and therefore cannot form a 3' to 5' bond. And the groin nucleic acid chain. Okay, so they're basically kind of telling us what. So they just want us to recall that like what typically happens in like an irregular formation between two deoxyribose molecules, and now they're just kind of giving a scenario where like where this is like this NRT, it prevents the formation of these bonds. So recall from like your content review, if we remember correctly, we have like our two deoxyribose, and so remember that deoxyribose is the one with one hydroxyl group, and then the ribose one has two hydroxyl groups. So I just want you to guys remember that and keep that in mind so you won't get mixed up. And so what happens here is that like they form this bond thing going on, and then like and that's a normal thing, and so they create this thing, and then that's just what normally goes on. But they're saying this NRTI this gets disrupted. It disrupts something, and so let's look at the and so they're saying like it's disrupting of the type of bond here. And so let's look at our answer choices. They're saying that it prevents the formation of the carbon to nitrogen bond. Now, if you go back to our our little diagram right here, I don't see no nitrogen at all because the formation between these bonds right here is through from a phosphate group and a oxygen group. This is the kind of bond you want to focus on right here. And so there's no nitrogen present. So as a result, this is not a correct answer. So we can eliminate A right away. Now they're saying it's B, carbon to oxygen. So now go back to again to the chart. They're saying, okay, this is a carbon right here to oxygen. So this is only talking about like this part right here. It's not really like focusing on this whole part right here for the other side. It's just focusing solely on like this previous carbon and oxygen. It's not really connecting to this other molecule. So it's not a good answer so far, even though it does look convincing if you don't haven't memorized the molecular structure for this. And so B is not a good answer. And then C, nitrogen to oxygen. Now again, it's like the same thing as answer choice A. There's no nitrogen present, so this eliminates C out, and this leaves D left. And so let me explain how the mechanism works. And so how this happens when like we have the hydroxyl group, which is from the deoxyribose, is nucleophilic. It's the nucleophile in this scenario. And so now it's gonna attack this phosphate group. So now what do you think it's gonna attack? It's gonna attack the phosphate, right? Because these oxygens, they are rich in electrons, so they're pulling the electrons like in their direction. So if the electrons are being pulled away from the phosphate, this makes the phosphate group a electron deficient. So this makes it pretty good candidate for being electrophile. So oxygen's gonna attack this phosphate group, and then it's gonna like kind of kick off one of its oxygens off to connect, and so we have this final formation bond right here. So in our final bond, it'll be an oxygen and a phosphate group because this oxygen connects to this phosphate group, and so we want this D to be our correct answer. And so if you didn't know how to solve this, you don't necessarily have to like memorize the structure of the DNA. You just have to understand and be familiar that when these two molecules are they're going through mechanisms to form each other. Just remember that it's going to be the oxygen and the phosphate group that are binding together. And so D is the correct answer. Question number 4. When compound 1 binds to the HIV protease, what is the value of this at equilibrium? So right away, this looks like a daunting equation, but this clicks to me. So like if you remember correctly from your content review, um, the formula for the constant of dissociation, it's when you have a free ligand or a free substrate, whatever you can call it, and then you have that free enzyme or protease. I'll just call this an enzyme for this example, over the binding complex. So when something's been disassociated, it's always going to be on the numerator that you're going to have the ligand and the free enzyme that are being dissociated from that binding complex. So whatever's being binded stays on the bottom, and then whatever is being like dissociated from that binding complex is on top of the numerator. And so then now, what if I were to take the reciprocal of this now? So now let's do flipped. So binding complex on top, and the others on the bottom. So now they're flipped, and now if you focus on this equation, notice that it's very similar to what we just saw in the question set. They said that the binding complex over the free compound, and then over like the unbounded protease. So this is very familiar with what we just took the reciprocal of. And so this equation here has a name. It's called the constant of association. It's the opposite of deassociation, in which these two ligands or like this enzyme and ligand, they bind together to create the binding complex. And so remember, and then the constant for association, the binding complex is always on the numerator, while in the dissociation, it's going to be in the denominator, so it's flipped. And so just note that and be familiar with these types of like equations for the MCAT. And so now if we go back, we want to find like the constant of association, because this is the definition of a constant of association. Now going back, they give us a constant of disassociation. We don't have a constant of association in the passage. They only give us KD. So what can we do? So like what we did in the problem right here, if we take the reciprocal of KD, we can find Ka. So let's do that. Let's take the reciprocal of this. So if they give us KD, which is 4 times 10 to the negative 3 nanometers. Is that correct? That is correct. So now, define Ka. We can do the reciprocal of Kd. So let's do that. Let's solve. So let's do 1 over times 4 times 10 to the 3 nanometers. Okay. So the only I'm going to do is probably better. I'm just going to probably keep the units out. So then we'll keep this as nanometers reciprocal. So we'll skip this out. Now, if we further break this apart, we get this 1/4 times 1 over 10^3. So then if you break this down, if you convert this to decimal form, know that for 1/4 it's a quarter, so it's going to be 0.25 times 1 over 10^3. Now, if you take the reciprocal of the exponent right here, if you move this on top, this becomes 0.25 times 10 to the third because of exponent rules. And so now if you were to convert this into an actual like number right here, this will be 250, and then we can bring our new units back here, and so this will be nanometers reciprocal, and so this should be our answer. And then this matches up for what we are solving right here. And so again, just to recap, remember that the KD association is the opposite of the constant association and vice versa. And so this is something you should be familiar with MCAT, and then that's the way you can solve this problem. And that is question number 4. Question number 5. Based on the passage, what is the most likely outcome of an LRA treatment on the viral load of an HIV-infected person that is also treated with ART? Now, a couple of questions ago, we discussed about this. I recommend quickly watching that just to get a fresh overview. But basically what they did was with this LRA treatment, their goal was to increase the initial amount of HIV so that the macrophages can produce antigens. And so when they produce antigens, then the immune system can respond. Because HIV has the ability to hide inside our genome, and then not be able to present those antigens because it's not being presented by the macrophages. And so they're basically asking this question: what will happen basically in this treatment? So I would predict that, like, if we were administered this LRS treatment, I expect a initial spike in HIV, right? Because the LRS, it promotes transcription of our genome, and so if it promotes transcription of the genome, it promotes the growth of like more HIV viruses. And so if there's more HIV being exposed, there's an increase of antigens, right? If there's an increase of antigens, the immune system responds because now it has something to go off from. So once it responds, then I expect a decrease in HIV. And so I expect like a graph like this, it goes up and then down like that. So now let's go back to our answer choices and see what fits best. So if we're looking at something that has a sharp increase and then it decreases later on. And so C makes the best answer choice because this is literally what we were talking about. We want a sharp increase followed by decrease after the immune system destroys the HIV. B and like D don't make any sense because B is saying a sharp decrease, we want increase, and then same thing right here is a decrease, we want to increase, and then between A and D and C, it's saying that it plateaus, and so it doesn't necessarily plateau because the immune system is gonna decrease the amount of HIV that's produced, and so it's not gonna plateau, it's gonna rather decrease, and so C is the correct answer for this question.